There's a rock on Mars, sitting in a dried-up riverbed that looks like something rotting. Not rotting the way meat rots, rotting the way an old riverbank rots when the water leaves and the mud sits there for years, going dark in blotches, iron bleeding into rings. On Earth, when you find a stone patterned that way, there's usually a boring reason and an interesting one. And the interesting one is that microbes lived and died in that mud. This particular stone is not on Earth, and the instrument that photographed those blotches was never really built to find them. That instrument is a laser on the end of a robotic arm on a rover called Perseverance, standing in Jezero crater. It fired an ultraviolet beam at the rock, read the light that bounced back, and reported carbon. Organic carbon. The kind with carbon locked to carbon in long, tangled chains. On the rock. Not deep inside it. Just sitting on the surface. In a place where the surface of Mars should have scrubbed it away 1 billion times over. And that's where this whole story stops being tidy and starts being a fight. If you find that kind of thing as strange as I do, this is the channel for it. Subscribe, hit like, and tell me in the comments what city you're watching from and what time it is where you are right now. I read those. Let's get into the rock. The rock has a name, Cheyava Falls. The team borrowed it from a waterfall in the Grand Canyon, and the borrowing is not an accident. The name, Cheyava, comes from the Hopi language and means intermittent waters. A waterfall that only runs when the snow melts and dries the rest of the year. They named a Martian rock after intermittent water because that is exactly what the rock records. Water that came, did its work, and left. White veins of calcium sulfate present clear evidence that water once ran through the rock. Picture the thing physically. It's about 1 meter long and a bit more than half a meter across, shaped roughly like an arrowhead lying in the open. Running the length of the rock are large white calcium sulfate veins, and between those veins are bands of material whose reddish color suggests hematite, one of the minerals that gives Mars its rusty hue. So already you have 2 stories written into one slab. The red bands are the old mud, iron-stained, laid down when this was the bottom of a river channel. The white veins are younger, cracks that opened later, and water pushed through them, carrying dissolved minerals that crystallized in place. 2 separate episodes of wet, frozen into stone. But nobody named the rock for its veins. They named it, and photographed it, and drilled it, because of the spots. Scientists were particularly interested in the millimeter-size, irregularly shaped light patches surrounded by a thin ring of dark material, akin to leopard spots. Pale in the middle, dark rim around the edge. Scattered across the reddish band like something spilled. And next to them, smaller and blacker, another texture the team started calling poppy seeds. The rock is peppered with black spots that the scientists call poppy seeds, and lighter spots with dark rims they call leopard spots. Here's why a geologist's pulse jumps at a spot like that. On Earth, that pattern has a chemistry behind it, and often that chemistry is life. Spotting of this type on sedimentary rocks can occur when chemical reactions involving hematite turn the rock from red to white. And those reactions can also release iron and phosphate, possibly causing the black halos to form. And they can be an energy source for microbes. Read that again slowly. The reaction that bleaches the red spot pale is the same reaction that could feed a bacterium. The spot is not a fossil. There's no little body in the middle. The spot is the leftover chemistry, the ash of a fire, not the fire. And a microbe eating iron in ancient mud leaves exactly this kind of ash. Two minerals sit inside those spots, and their names matter, so here are both. The leopard spots carried the signature of Vivianite, a hydrated iron phosphate, and Greigite, an iron sulfide. Vivianite is a blue-green iron phosphate. On Earth, you dig it up in swamps, in peat, around things that died and rotted in wet ground. Vivianite appears within the black spots the team calls poppy seeds, and in the dark rims of the larger leopard spots. Greigite is an iron sulfide, and on Earth certain microbes manufacture it as a byproduct of how they breathe. Vivianite is frequently found on Earth, in sediments and peat bogs, and around decaying organic matter. Similarly, certain forms of microbial life on Earth can produce Greigite. So put the picture together. Old river mud, organic carbon in it, iron, sulfur, phosphorus, and spots arranged in a bullseye. Vivianite in a rim, Greigite in a core, that on Earth would make an astrobiologist reach for the phone. The Martian rock shows rims rich in Vivianite, surrounding small cores enriched in Greigite, and that bullseye pattern matches a sequence of electron transfer reactions seen in some Earth sediments. And here's the caution, before anyone gets ahead of the evidence. Because a molecule made of carbon is one of the most oversold things in all of science. Finding organic carbon on Mars sounds like finding a footprint. It is not. It's finding a rock that could, in principle, have been walked on. The word organic fools people. It sounds like it means made by an organism. It doesn't. Organic does not necessarily mean formed by living things. It just means having a lot of carbon-carbon bonds, and there are other processes that can make those besides life. Comets carry organic carbon. Meteorites carry it. Hot water reacting with plain rock cooks it up out of nothing biological at all. Macromolecular carbon is a class of large, cross-linked carbon networks found in fossilized biological carbon on Earth, in meteorites, and in rocks formed through abiotic hydrothermal reactions. Same tangle of carbon, three completely different origins. One of them boring, one of them cosmic, one of them alive. The molecule itself will not tell you which. That is the whole cruelty of this search. The most exciting thing you can find is also, by itself, worthless. Which is exactly why the second half of this rock matters more than the carbon. It's not that Cheyava Falls has organic carbon, it's that the carbon sits right next to those minerals, and the minerals are arranged in a pattern, and the pattern is the kind of thing electron-shuffling chemistry leaves behind. It's not just the minerals, it's how they are arranged in these structures that suggests that they formed through the redox cycling of iron and sulfur. On Earth, that arrangement shows up where microbes are eating carbon and, in a manner of speaking, breathing rust and sulfate instead of oxygen. Organic carbon gives up its electrons, iron and sulfur take them in, and the leftover minerals precipitate in rings around the reaction. On Earth, things like these sometimes form in sediments where microbes are eating organic matter and breathing rust and sulfate. Now step back and appreciate how long we waited to even be allowed to say a sentence like that. Because for most of the history of Mars exploration, the word life was, in effect, banned. It goes back to 1976. Two landers named Viking touched down on Mars, and unlike everything that came after them, they carried instruments built to do one thing: test the soil directly for living things. The 1976 Viking landers remained the only missions to explicitly test for extant life on Mars. One of those tests was called the Labeled Release Experiment, and the idea was almost embarrassingly direct. The experiment took a sample of Martian soil, applied a drop of nutrient solution tagged with radioactive carbon-14, and radioactive carbon dioxide would then be detected only if life were present. Feed the soil. If something metabolizes the food, it exhales tagged gas. Simple. And the soil exhaled. One experiment, the labeled release experiment, showed that the Martian soil tested positive for metabolism, a sign that, on Earth, would almost certainly suggest the presence of life. Both landers, 4,000 miles apart, gave the same repeatable positive response. It should have been the biggest news in the history of the species. It wasn't, because a second instrument on the same lander went looking for the actual organic molecules that any living thing would leave behind, and found nothing. A related experiment found no trace of organic material, suggesting the absence of life. So Viking left the human race with a stalemate. One instrument said, "Something's alive down here." Another said, "There's nothing here to be alive." Overall, these life detection experiments produced surprising and contradictory results. Most scientists eventually decided the positive signal was a chemical trick, some aggressive oxidizing compound in the soil that fakes metabolism by tearing the nutrient apart on contact. Not everyone agreed, and the man who designed the experiment argued to the end of his life that Viking really had found life. But the consensus settled on chemistry, not biology. Both landers reported positive results when the soil was tested for the presence of microbes. But most scientists have concluded that those results were caused by unusual chemistry in the soil, not life. And that ambiguity did something you might not expect. It scared NASA off the entire question. There followed an explicit ban of decades on such experiments, imposed immediately after the 1976 Viking mission, and the agency attributes the ban to the ambiguity of that mission's labeled release experiment, fearing an adverse effect on the space program should a similar inconclusive result come from a new robotic quest. Think about that. A result so muddy it wasn't a no and wasn't a yes was frightening enough that the safest institutional move was to stop asking. For decades, the rovers that followed weren't sent to find life. They were sent to ask a smaller, safer question. Could Mars ever have supported life at all? They are focused on past habitability, whether or not Mars could have supported life in the past. Curiosity, the rover before Perseverance, officially carried no life detection experiment at all. That's the wall Cheyava Falls runs into. Half a century of being burned by ambiguity, and a whole vocabulary built to never overpromise again. So when the carbon signal came back from Jezero, the first thing the team did was try to prove it wasn't real. Not celebrate it. Attack it. And the attacks were specific, because there were obvious ways to be fooled. The rock the carbon was found on lives at a site called Bright Angel, and Bright Angel had a complication. It was the first site the instrument examined after a dust cover anomaly disabled its focusing mechanism, forcing the team to adopt a new operating mode. A broken focus is exactly the kind of thing that manufactures a fake signal. The first fear was almost comically mundane. The instrument fires its laser through a little window of fused silica, and glass itself, hit with ultraviolet light, can glow in ways that mimic organic carbon. The first concern was that the signal could have been light bouncing off the instrument's own fused silica front window. The carbon might not be on Mars at all. It might be on the lens. So the team took spare copies of the optics back in a lab on Earth, fired lasers at nothing in particular on Mars, aimed at known blank targets, and pointed the instrument at an ordinary neighboring rock they'd chosen as a control, a rock called Steamboat Mountain. The final confirmation came when the team pointed it at Steamboat Mountain. Other rock targets nearby do not exhibit the carbon spectral signal. The Bright Angel signal was not coming from hardware. If it were the window, every rock would glow. Only some did. The signal was in the ground. The second fear was worse, in a way: contamination. What if the rover carried the carbon from Earth and smeared it onto Mars? A rover assembled in a clean room is still assembled by people in an atmosphere full of Earth's chemistry. That one takes more than a control rock to rule out, and it's part of why the team keeps insisting the real answer is locked up in a sample tube waiting for a lab. But they cleared enough of the alarms to say something out loud, and in September 2025, they said it. A sample collected by Perseverance from an ancient dry riverbed in Jezero crater could preserve evidence of ancient microbial life. Taken from Cheyava Falls, the sample, called Sapphire Canyon, contains potential biosignatures, according to a paper published in the journal Nature. Sapphire Canyon is the core they drilled out of Cheyava Falls, sealed in a metal tube, sitting on Mars right now. And the phrase they chose, potential biosignature, is a phrase engineered by 50 years of getting burned. A potential biosignature is a substance or structure that might have a biological origin, but requires more data or further study before a conclusion can be reached about the absence or presence of life. Now, here is the part that I find almost unbearable, and it's the reason this rock is not a headline that ends the search, but a knife that cuts both ways at once. The same paper that called Sapphire Canyon a potential biosignature also, in effect, handed the skeptics their weapon. Because those two minerals, Vivianite and Greigite, the whole reason the spots look biological, can be built with no life at all. The team knew it and published it anyway. On Earth, Greigite forming from sulfate and organic matter is a reaction so sluggish at ordinary temperatures that you basically never catch it happening on its own. The reaction between sulfate and organic matter at room temperature is incredibly slow. So slow that we don't actually observe it happening in natural environments on Earth. Where we do see it is where biology is involved. That's the case for life. Living cells are the only thing patient and efficient enough to drive that reaction in cold mud. Except there's a second door. The other place this can happen is if you take sulfate and organic matter and cook them together at temperatures above about 120 to 140 degrees, overcoming the kinetic inhibition that exists at lower temperatures and driving the production of reduced sulfur-bearing minerals. Heat it hot enough, and dead chemistry makes the identical mineral. So the argument becomes a fight over temperature. Cold, and life is the best explanation. Hot, and you don't need life at all. The spots look the same either way, and the Vivianite is even easier to fake. One of the lead scientists said it plainly. The iron phosphate side of the reaction feels like the kind of thing someone will eventually reproduce in a beaker with no biology involved. Ferric iron and organic matter are quite reactive toward one another, and so the redox reactions that make Vivianite, it feels plausible that someone will figure out that that can be done completely abiotically. And billions of years is a long time to run a slow reaction. Because these Mars rocks are billions of years old, that offers abundant time in which the speckles could conceivably have emerged from abiotic processes. Small pockets of reduced Vivianite and sulfides formed long ago by run-of-the-mill chemical reactions, rather than by hungry microbes. So read what actually arrived in that one paper. The best evidence for ancient life ever found on Mars. And, in the same document, the exact recipe for making that evidence with no life in it. The signal and its own alibi delivered together in the same breath by the same team. They found the most compelling case they've ever had, and they were honest enough to hand you the reason it might be nothing. What's left of the claim, once you've admitted dead rock can forge it? That question is the thing standing between us and an answer. And it turns out the people who found the rock have a very precise, almost brutal way of scoring exactly how far from life they still are. Life can hide inside cold water. That's the whole trap of Sapphire Canyon. And once you understand it, you understand why the team couldn't just declare victory. On Earth, in cold, wet mud, the reaction that builds those spots is so lazy, so reluctant to happen on its own, that when you find it finished, you assume something living pushed it along. But dead water has a second way in, and the second way is worse than useless. It makes an identical rock. So the question is not whether Cheyava Falls looks like life. It does. What matters is whether the mud was cold. And a rock can't tell you the temperature it sat in 3.5 billion years ago. Watch how the imitation works, step by step, because the mechanism is the whole argument. Take iron in its rusted, oxidized form. Ferric iron. Iron that has already given away electrons. Put it next to organic carbon. Carbon still loaded with electrons to spare. Those two are chemically restless toward each other, the way a stretched spring is restless. The organic carbon wants to hand its electrons off. The ferric iron wants to take them. When that transfer happens, the iron flips to a reduced state, and reduced iron combines with phosphate dissolved in the water to precipitate as Vivianite, that blue-green mineral that on Earth grows around things that rotted in swamps. Nearby, sulfate takes electrons too and drops out as Greigite, the iron sulfide. Ring of one mineral, core of the other. The leopard spot. Now here's the cruel part. Nothing in that chain requires a cell. It requires iron, carbon, phosphate, sulfate, water, and time. A microbe speeds it up enormously. A living thing is a machine for pulling electrons off carbon and shoving them onto iron and sulfur. That's literally how some bacteria breathe. But strip the microbe out, and the reaction still runs. It just runs slowly. And slowly, across 3.5 billion years, is not slow at all. Give ordinary dead chemistry that much time, and it will grind through reactions that, on a human clock, look frozen. The rock had the time. That's the alibi. Small pockets of reduced iron and sulfide, forming grain by grain over eons. No biology anywhere. There's an even simpler crack in the case, and it's temperature. The one thing that would settle it, was this mud cold or hot, is exactly the thing the rock refuses to reveal. If the water was cold, life is the front runner, because cold, dead chemistry is too sluggish to explain finished spots, and life is the patient exception. But heat the same ingredients past the point where water would boil at Earth's surface, and the reluctant reaction stops being reluctant. Dead chemistry catches up, makes the same Vivianite, the same Greigite, the same bullseye. And Jezero, billions of years ago, had volcanic rock and probably had warm water moving through cracks. So both stories fit the same stone. This is the corner the team is painted into. The more carefully you look at the spots, the more they insist on staying ambiguous. So why publish the doubt right next to the discovery? Why hand the skeptics the weapon in the same paper? Because of what happened the last time a Mars team didn't. In 1996, a group announced that a meteorite from Mars, found in Antarctica, contained microscopic structures they read as fossil bacteria. It made the President of the United States speak on television. And then, over the following years, every one of those structures got explained without life. The tiny shapes were too small to be cells. The minerals could form without biology. The organics could have crept in from Earth. The claim didn't just fade. It became a cautionary tale taught to every young scientist in the field. A lesson in how a beautiful result can be picked apart in public and take a reputation with it. A generation grew up watching that. So when this team found something real, they front-loaded every reason it might be wrong. Not out of modesty, out of memory. Publishing your own strongest counterargument is how you survive the pattern that ate the last people who got excited. That's why the honesty reads as strength. A team that lists the ways it could be fooled is a team that has thought harder than anyone about being fooled. Which brings us to the strangest instrument in this whole business. And it isn't the laser or the drill. It's a ruler. The mission uses a scale, 7 steps, to grade how confident anyone is allowed to be about a possible sign of life. And it is not a scale of feelings. It's closer to a scale of distance. Distance between we saw a hint and there was life. The scale runs from the bottom, where you've merely detected something that could, in principle, be biological, all the way up to the top, where you've ruled out every non-living explanation and confirmed life beyond argument. Level 1 is the very first rung. You found a possible biosignature, and you can't yet rule out that your own instrument made it. Remember the fear that the carbon might be glowing off the laser's glass window? That's a level 1 problem. As you climb, each rung demands you kill another alternative. Prove it's really on Mars, not contamination. That's a higher rung. Prove the geological context makes sense. Higher still. Prove no dead chemistry can fake it. Higher again. Establish it's genuinely biological in origin. And near the very top, do it all with a sample examined by many independent labs, so no single mistake can carry it. So where does Cheyava Falls sit? Low. Somewhere on the bottom rungs. The mission has confirmed a possible biosignature that is really on Mars and really in ancient rock, which clears the first hurdles, but it has not, and cannot with the instruments on the rover, kill the abiotic alternative. It is stuck at the exact rung where the alibi lives. And here's the thing that stops me cold when I actually sit with it. Nothing ever found on Mars, in the entire history of looking, has climbed higher than these lowest rungs. Not Viking, not the methane, not this rock. Every candidate in 70 years of searching has stalled in the bottom third of the ladder, because every candidate ran into the same wall. A dead explanation that fit the data just as well. Here's what a low rung really means. Because it's easy to hear level 3 and picture a number on a chart. It isn't a chart. It's a rock, in a sealed tube, on the floor of a crater, on another planet, that we cannot open. When the scale says Cheyava Falls sits on a low rung, the real meaning is that the evidence to move it up isn't hidden in the data. It's hidden in the sample, which is 100 million miles away and going nowhere until someone flies it home. The rung isn't a measure of the rock, it's a measure of our reach. We found the best case for ancient Martian life yet discovered, and we placed it near the bottom of our own confidence scale. Not because it's weak, but because we're too far away to make it strong. That's the honest translation. Low rung means we can see it, and we can't touch it. To understand why touching it matters so much, you have to go down to the single atom, to the carbon itself. Because that is where the cleanest fingerprint of life actually lives. And it is a fingerprint you cannot read from Mars. Here's the trick. Carbon comes in 2 stable weights. Most of it is the lighter kind. A small fraction is a heavier version, one that carries a couple of extra neutrons and drags a little more when it moves. To any ordinary chemical reaction, The two are nearly identical. Same bonds, same behavior. But not to a living cell. A cell is a molecular machine, and when it grabs carbon to build itself, its enzymes reach a hair faster for the lighter atoms, because lighter atoms move a hair faster and slot into the machinery more readily. Do that trillions of times, in cell after cell, generation after generation, and the living matter ends up measurably enriched in light carbon, while the heavy carbon gets left behind in the environment. Life sorts atoms by weight. It can't help it. The sorting is a side effect of how enzymes work. A rock, by contrast, doesn't sort. Dead chemistry treats the two weights almost the same, so carbon cooked up by hot water or delivered by a meteorite carries roughly the ratio it started with. So there's your test, the sharpest one we have. Measure the ratio of light carbon to heavy carbon in the organics. If they're strongly skewed toward light, that's the signature of a machine that was choosing. And this is not theory. It's how we found the oldest evidence for life on Earth. In some of the most ancient rocks on this planet, older than any fossil, there's no body, no shape, nothing you'd recognize as a creature, just carbon, with the light isotopes overrepresented, the leftover fingerprint of cells that sorted their atoms and then vanished, leaving only the arithmetic of their preference behind. That fingerprint is the thing we cannot yet read from Cheyava Falls. Not because it isn't there, it may be, sealed in Sapphire Canyon, but because the instruments, precise enough to measure an isotope ratio to the needed accuracy, don't fly on a rover. They fill a room. They need a sample in hand, prepared, run, and rerun. That measurement is the whole reason the sample is in a tube instead of a headline. The carbon on Mars is either whispering, "I was chosen," or "I was random." And the only way to hear which is to bring it home. Except time has been at work on that whisper, and time on Mars is not kind to a fingerprint. Consider what 3.5 billion years of Martian surface does to an organic molecule. Mars has no magnetic field to speak of, and only a wisp of atmosphere, so the ground is bathed in radiation. Cosmic rays from deep space and charged particles from the sun, day after day, eon after eon. Radiation doesn't preserve delicate carbon chains. It breaks them. Every hit snaps a bond, scrambles a structure, degrades a complex molecule into something simpler and more anonymous. On Earth, a body gets buried and shielded. On Mars, the top layers of rock have been sitting in the beam for longer than there's been complex life anywhere we know of. So the signal doesn't vanish all at once. It smears. A molecule that was once a specific, informative structure, the kind that shouts "biology," gets ground down into generic, tangled carbon, the kind that says nothing. The isotope ratio is tougher. Radiation doesn't easily change which atoms are heavy and which are light, so that fingerprint survives better than the molecular shapes do. But even that gets muddied, because radiation and time mix things, add contamination, blur the original ratio with later chemistry. What you're left with, after 3.5 billion years, is not a proof. It's a rumor of a proof. A signal degraded to the point where it's consistent with life, but no longer able to demand it. And that's the deeper problem. Deeper even than not being able to reach the sample. The sample itself has been slowly erased for longer than the human lineage has existed. And what remains may be a signal that can survive only as a suggestion of itself, never sharp enough to be undeniable, no matter how carefully we look. The rock kept the mud, kept the veins, kept the spots. What it may not have kept, in full, is the one atomic-scale detail that would settle the whole argument. And if you think that's a discouraging place to leave it, there's a second candidate for life on Mars that has haunted this search even longer than the rocks, and it behaves in a way no rock ever could. It moves, it comes and goes, and it nearly drove the people chasing it out of their minds. It's methane. A gas, the simplest one a living thing can breathe out. On Earth, the overwhelming majority of methane is made by life, by microbes in mud, in guts, in the deep subsurface, exhaling it as waste. So when Curiosity, the rover working in a different crater called Gale, began sniffing tiny amounts of methane in the Martian air, it lit up the whole field. Because methane on Mars is a paradox before you even ask where it came from. Sunlight and chemistry destroy methane in the Martian atmosphere within a few centuries, a geological eye blink. So any methane you detect can't be ancient. It had to have been released recently, and something has to keep releasing it, or it would already be gone. Then it got stranger. The methane wasn't steady. Curiosity found that the levels rose at night and fell away by day, and drifted with the seasons, peaking in warmer months. A gas that pulses, that behaves almost like breath. Night after night it would climb, and by Martian morning it would thin back down toward nothing. Something was letting it out under the cover of dark and pulling it back, or destroying it once the sun came up. And that's where the ghost turned truly maddening. A spacecraft in orbit around Mars, a probe built specifically, exquisitely, to detect trace gases from above, far more sensitive than the rover, flew over the same region and looked for that methane in the air column. And it found nothing. Not a reduced amount. Nothing. Below its detection limit, again and again. So you had a rover on the ground, swearing it smelled methane, and an orbiter overhead, swearing the air was clean. Two of the best instruments ever sent to another planet, over the same crater, flatly contradicting each other. For years, that contradiction sat there, unresolved. A gas that existed at the surface and didn't exist in the sky. It took a long time to even partly untangle, and the answer, when it came, was humbling. Part of the methane, it turned out, wasn't Martian in the way anyone hoped. The rover itself was implicated. Its own wheels grinding across the surface, crushing rock, may release trapped gases. And gas can leak from within the instruments and machinery as they operate. Some of the ghost was the machine. And the rest of it, the part that pulses at night, points to methane seeping up from below the crater floor, trapped in the ground and released as temperature and pressure shift through the Martian day, then flattened out and diluted in the daytime air so thoroughly that an orbiter scanning the whole air column would never see the faint concentration hugging the ground at night. The orbiter wasn't wrong. The rover wasn't wrong. They were measuring the same gas at two scales, and at the orbiter's scale, it genuinely averaged out to nothing. So the great methane mystery got cut in half and handed back in two unsatisfying pieces. Half machine, the rover contaminating its own search. Half geology, gas leaking from the rock, released by the daily thermal squeeze of the crust. No life required. The ghost was part hardware, part plumbing. And notice what that does and doesn't settle. It explains the contradiction. It does not explain the gas. Even the geological half leaves the deeper question open. What's making methane down there in the first place? Because underground, gas can be produced by water reacting with hot rock, no biology anywhere. Or it can be made by microbes, exactly the way it is on Earth, sheltered from the killing radiation up top, still exhaling into the dark. The two sources make the same molecule. We are right back at the wall. But the methane did teach us one thing that quietly rewrites everything else in this story. And it's this. For all the years we spent treating Mars as a dead, finished, frozen museum, a planet whose interest in chemistry ended billions of years ago, where the only question was what it used to be. The gas seeping out of Gale crater at night is happening now. Not in the ancient past. Tonight. Something under the surface of Mars is still releasing gas, still driven by heat and pressure moving through the ground, in a place where liquid water may still exist far below. The planet we called geologically dead is, at least in its plumbing, quietly alive with process. And that changes the whole shape of the search. Because if the interest in chemistry isn't only locked in 3.5 billion-year-old rock, but is still running underground, out of the radiation, in the dark, then we've been looking in the wrong place. We've been reading the ancient surface like a gravestone. The thing worth finding may not be up here at all. What that redirection actually means is uncomfortable. So let me say it plainly. The most interesting rover on Mars keeps sending home pieces of a puzzle nobody drew. Perseverance was built to answer a modest question. Was Jezero ever habitable? And instead it keeps finding things that don't fit the question at all. Spots that could be biology, or could be dead chemistry. Carbon that could be a fingerprint, or could be noise. And the more it looks, the less the surface behaves like a settled record, and the more it behaves like an instrument still taking readings of a planet that hasn't stopped changing. Mars stopped being the backdrop of this story. It became the thing under the microscope, and the microscope keeps flinching. Which forces a question that sits underneath everything. How much of what we found on Mars was actually Mars? And how much was the machine looking at itself? Because this has happened before, more than once, and the pattern is its own kind of warning. Remember the first fear about the carbon on Cheyava Falls? That it might be light bouncing off the instrument's own glass window, not a signal from the rock at all. That fear wasn't paranoid. It was learned. The history of Mars exploration is littered with signals that turned out to be shadows of the equipment. The methane we spent this whole story on, a piece of it was the rover's own wheels grinding rock, gas leaking from its own machinery as it worked. The ghost that pulsed at night was, in part, the ghost of the thing sent to hunt it. Go back further, and the pattern deepens. When Viking's soil test came back positive for metabolism in 1976, the leading explanation wasn't life. It was that the Martian soil contained aggressive oxidizing compounds that tore the nutrient apart chemically, faking the exhale of a living thing. In other words, the soil was reactive in a way the experiment couldn't tell apart from biology. The instrument was designed to catch life, and it caught chemistry wearing life's coat. The whole reason NASA banned direct life detection experiments for decades afterward was this exact terror, that any positive signal would be indistinguishable from the instrument being fooled. Even the water, the thing everyone agrees Mars once had, has had its illusions. Dark streaks on crater slopes were read for years as possible flowing brine, seasonal trickles of salty water. Then closer study suggested many of them were dry, grains of sand and dust sliding downhill, not liquid at all. A planet's most famous water features reinterpreted as avalanches of dust. Not because anyone lied, but because at the edge of detection, a dead process and a living hope can produce the same picture, and the human eye reaches for the hope. So when the rover reports leopard spots and organic carbon, understand what a fragile sentence that is. Every one of those findings has had to survive a gauntlet. Is it contamination from Earth? Is it the instrument's glass? Is it the wheels? Is it noise? Is it the machine's own shadow in the data? The team clears what it can, but some of the shadows can't be cleared with a rover at all. And that's the honest limit of trying to read a planet through a robot's senses from 100 million miles away. Not a flaw in the rover. The instrument is always, faintly, in the picture. And then Mars does something that no shadow of a machine can explain. It hands you a rock that shouldn't exist. On a ridge the team was crossing in Jezero, Perseverance drove over a pale stone and cracked it open under its wheel. And inside was yellow crystal. Pure, elemental sulfur. Not sulfate, not the sulfur locked into salts that Mars has everywhere. Pure, native, crystalline sulfur, the bright yellow kind, sitting inside a broken rock. The rover accidentally cracked open a rock, revealing yellow crystals of elemental sulfur, something never before seen on Mars. Here's why that stops geologists in their tracks. Sulfate, sulfur bound to oxygen and metals, is ordinary on Mars, expected, understood. Pure elemental sulfur is something else entirely. On Earth, native sulfur forms in specific, narrow conditions, around volcanic vents, in hot springs, and, this is the part that matters, as a byproduct of certain microbes that make their living off sulfur chemistry. And here's the deeper problem. None of the geological processes we know were active in that part of Mars are supposed to produce pure sulfur there. Elemental sulfur forms only in a narrow range of conditions that scientists don't associate with this location's history. The rock was found in a field of similar-looking rocks, which means it isn't a fluke, isn't one weird crystal. There may be a whole region of this stuff. And the process that made it isn't in the playbook for that place. So Mars produced, at scale, a mineral that the local geology has no accepted recipe for. And this could never be a contamination artifact. Pure sulfur crystals can't be smeared onto Mars from a clean room on Earth. The rover cracked a rock and found the planet doing something we can't yet explain, using chemistry we didn't know was running there. The object is real. Its cause is missing. The explanations offered so far don't hold, and it's worth saying them flat, because watching each one fail is how you understand the size of the gap. One idea: ancient hydrothermal water, hot fluid moving through rock, could concentrate sulfur. But the evidence for that kind of hot system in that specific spot at the right time is thin. Another idea: some low-temperature chemical pathway we haven't characterized. Dead reactions in cold water over long timescales. The same "give it billions of years" argument that shadows the Leopard spots. Maybe. But nobody can point to the reaction and say, "Here, this is it." And a third possibility hovers unspoken, because sulfur is one of the elements life on Earth builds its metabolism around. The same uncomfortable awe that follows every finding in this story. Life makes native sulfur here. So could Mars have. And once again, the mineral looks the same, regardless of which door it came through. None of these can be tested. That's the wall, and it's the same wall every single time. Was the water hot or cold? The rock won't say. Is the carbon light or heavy? The rover can't measure it finely enough. Did microbes make the sulfur, or did dead chemistry? You cannot tell from a photograph and a coarse reading in the field. Every one of these questions has the same answer. Not until the rock comes home. The rover has taken us exactly as far as a robot can go, and set us down at a locked door, holding the key in a place we can't reach. The key is 30 tubes. That's the plan, and it's already partly done. Perseverance has been drilling cores as it goes, finger-sized cylinders of Martian rock. sealing each one in an ultra-clean metal tube, and either caching them on the ground or carrying them aboard. Sapphire Canyon, the core from Cheyava Falls with the potential biosignature, is one of them. The idea was elegant and enormous. A later mission would come, gather the tubes, launch them off the surface of Mars, the first launch from another planet in history. Rendezvous with an orbiter and fly them back to Earth, where the room-sized instruments that can read an isotope ratio to the needed precision are waiting. And then the arithmetic collapsed. The return mission's projected cost climbed past what anyone was prepared to pay. The schedule slipped past a decade, and the whole architecture was pulled back for redesign. Recent budget proposals moved to cancel the sample return effort outright. As it stands, there is a real chance the machine built to fetch those tubes never gets built at all, which leaves the tubes exactly where they are. Some sealed on the rover, some already dropped in a careful pile on the crater floor, a small depot of Martian rock arranged by a robot for a spacecraft that may never come. I want to be careful not to dress that up as tragedy, because it isn't one. It's just a fact with a strange shape. There is, sitting on the ground of another planet right now, quite possibly the best physical evidence for life beyond Earth that our species has ever collected. It is drilled, sealed, catalogued, and waiting. And the thing standing between it and an answer is not distance, not physics, not the erosion of billions of years. It's a budget line. The samples that could move Cheyava Falls up that seven-rung ladder are ready. The reach to grab them is the thing in doubt. After 70 years of asking whether Mars ever held life, we finally have candidates worth the argument. And they may simply stay there, in the cold, unopened, because the return trip is expensive. That's not a cliffhanger. That's the actual situation. But suppose the tubes do come home. Suppose the money appears, the rocket launches, the samples land in a sealed lab. Here is the harder truth, the one that takes the shine off the rescue. They might still not settle it. Because we can't even agree about our own planet. Some of the oldest evidence for life on Earth, the light carbon fingerprints, the tiny structures in ancient rock, is still argued over by serious scientists, decades after the samples were pulled from the ground and handed to the best labs on the planet. Those rocks are here. We can touch them, slice them, run them through any instrument we own as many times as we like. And still there are careful people who look at the same 3.5 billion-year-old carbon and say, life. And other careful people who look at it and say, you haven't ruled out dead chemistry. If we can't reach consensus on the oldest life in rocks we've held in our hands for 40 years, what exactly do we expect to prove from a Martian core degraded by even more radiation from an environment we understand far less? This is the gap that turns out to be the whole game. The gap between habitable and inhabited. Perseverance's original mission, the safe question, was habitability. Could Jezero have supported life? And the answer to that is a confident yes. There was a lake. There was a river delta. There was liquid water, the right minerals, chemical energy, long stretches of time. Gale Crater, where the other rover works, tells the same story. An ancient lakebed, a place life could have lived. We have proven, thoroughly, that Mars was once a world where life was possible. That was the achievable goal, and it's done. But possible is not the same as present. A habitable world is an empty room with the lights on and the door open. It tells you a guest could have come. It doesn't tell you one ever did. Every finding in this story lives in that gap. The carbon, the spots, the sulfur, the methane. Each one is consistent with a guest having been there. Not one of them proves the room was ever occupied. And the entire difficulty of Martian biology is that the room has been empty and irradiated for so long that even if a guest did come, the evidence they left may have decayed past the point of certainty. We might close the gap. We might also find that the gap can't be closed with what remains. So let me take you back to those leopard spots. The pale centers, the dark rims, scattered across the red mud of a rock named for water that comes and goes. When I first described them, I said a geologist on Earth would reach for the phone. And that's true. But sit with what we've learned since, and the meaning of "we found something" changes underneath you. We found a pattern. The pattern is real. It's on Mars. It's in ancient rock. It survived every test the rover could throw at it. What we did not find is what made the pattern. And there may be no way from here to ever know. That's a stranger place to end than "we found life" or "we found nothing." Because it means the thing we're actually confronting isn't a fact about Mars. It's a question about proof itself. About what it takes to be sure of anything that happened 3.5 billion years ago on a world we can barely touch. The leopard spots don't answer whether Mars had life. They ask us how sure we're allowed to be. And then they refuse to help. And so, the tubes wait. Sealed metal cylinders on the floor of a crater, in a thin, cold atmosphere, under a sky that never quite goes black at midday, holding cores of rock that might carry the oldest secret in the solar system. Or might carry nothing but patient, dead chemistry that learned over eons to look exactly like a secret. They will keep for a very long time out there. The cold preserves them. Whether they last was never the worry. The real question is whether we'll go, and whether, when we finally hold them, we'll be able to agree on what we're looking at. For now, that's where it stands. The best answer we've ever found to the oldest question we've ever asked. Drilled and sealed and set down gently on another world, waiting to see if we ever decide it's worth coming back for. If this is the kind of question you want to keep sitting with, subscribe and stay with the channel. There's a whole universe of these still unopened.